Hello everyone, I am Anushua Biswas and today I'll be presenting our work resolving diverse protein DNA footprints from exonuclease-based chip experiments. We know transcription factors bind to the DNA to influence transcriptional regulation. They may be binding directly to the DNA or as part of complexes with other proteins. Identifying these genomic binding sites is crucial to understand their roles in gene regulation. One of the most common methods to identify the protein binding sites is chromatin immunoprecipitation. In chip, formaldehyde is used to first cross-link the proteins to the DNA. Then the DNA is sheared into fragments of average size 200 to 1000 base pairs. After that, a specific antibody is used to immunoprecipitate the chromatin associated with the target protein. Then the crosslinks are reversed and these DNA fragments are mapped to the genome. We see a large number of these DNA fragments or reads near the protein binding location, forming peaks. In between the peaks on the forward and the reverse strand, we get a broad region, but the protein actually binds to a much shorter region of around 20 base pairs. A modified version of the Chipexo protocol is the Chipexo protocol. The first few steps are similar to that of ChIP-seq. After immunoprecipitation, it is treated with a lambda exonuclease enzyme that ties the free DNA from 5' to 3' end till it encounters some hindrance in the form of the bound protein. When these DNA fragments are mapped to the genome, we see sharp, precise read distributions very close to the actual protein binding sites. However, these Chipexo footprints are not the same at all the locations for a profiled protein. These Chipexo footprints differ at different locations and it depends on many factors, namely the actual protein binding to the DNA. It can be binding directly or as part of a complex with other proteins. The actual nucleotide sequence to which the protein binds. These may differ slightly and the protein might be binding with different binding affinities. The precise location of the crosslink within the protein DNA complex. These give rise to variations in the motifs as well as the read distributions, and a single overrepresented motif will not be able to explain the entire dataset. Currently, there are two broad approaches to categorize these Chipexo footprints. Exoprofiler pipeline by Static et al. They use known DNA motifs to scan the regions for putative transcription factor binding sites. Chipexo footprints are plotted and analyzed separately at these sites to identify the potential complexes forming there. The other approach is ChexMix by Yamada et al. They first initialize the potential binding regions into subtypes based on either de novo motif finding or on the distinct read distributions detected in the top most enriched regions. After that, they use EM to refine these subtypes over the complete dataset. We present our method Exodiversity. It is a model-based Bayesian framework that uses both sources of information, the read distributions as well as the DNA information, simultaneously to partition the dataset into different modes of protein binding, with each mode having its own characteristic motif and the respective read distributions. Let us look at the input, model, and the working of our algorithm next. The input to exodiversity is in the form of three datasets. A set of DNA sequences X, which is typically the only input given to a motif finding algorithm. But here we also have the positive strand read counts and the negative strand read counts as R plus and R minus corresponding to these DNA regions. They are then converted into binary format. Here we can clearly see a signal at the center, which is the summit of the peaks. The question which exodiversity wants to answer is there a single type of footprint or are there multiple ones? And can we identify these multiple footprints only using these three datasets? Indeed, exodiversity divides the ChIP-Nexus SOX2 dataset into 12 modes of binding, with each mode having its own characteristic motif and positive and negative strand read distributions. This is the same data we saw before, just reordered and aligned according to the mode positions identified by exodiversity. We now look at the model. Consider a data point xi, ri plus, ri minus. The parameters of the model include the mode of the data point ii, which is equal to k here, the starting position of the motif, which is zi. The motif is modeled as a PSSM phi k. At a fixed distance tau k plus, we have the positive strand read window. It is modeled as Bernoulli distributions over 0 and 1 at each position of the read window. Similarly, at a fixed distance, tau k minus, we have the negative strand read window. It is also similarly modeled and is denoted as phi k minus. Now, given a data point, the likelihood of it is calculated as a product of the probabilities P of xi given theta m and P of ri plus given theta m and P of ri minus given theta m. These probabilities are independent given the parameters of the model. All these parameters are unknown. Our goal is to learn the values of these parameters by sampling from the posterior distributions to maximize the posterior probability. The posterior probability is given as proportional to the likelihood times the prior. We will first look at the sampling of the motif start position. Consider three sets of sequences x, r plus and r minus belonging to a mode k. Starting from some initial value of wk, which is the width of the motif, and tau k plus and tau k minus, we randomly initialize the motif start positions in x and correspondingly determine the position of the positive and the negative strand read windows. In the next step, we remove a sequence xi and the corresponding sequences ri plus and ri minus. From the remaining sequences belonging to the same mode, we calculate our PSSMs phi k plus and phi k minus. Given these and the fixed values of tau k, we calculate a score for every possible position of the motif. From this score vector, we sample for the motif start position. Let it be zi equal to j here. Once we know the motif start position, we can easily determine the position of the positive and the negative strand read windows. Keeping the motif start position fixed, we now sample the mode for the data point. Let us consider we have three modes. For each mode, we calculate a score from the three components in ri plus, ri minus and xi. Note that for each mode, the value for the offset tau can be different for both the read footprints. From this vector, we sample the mode of the data point. Let it be ii equal to 1 here. In this way, we sample for the motif start position and the mode for all the sequences. And after that, we also sample for the width of the motif and the value for tau k plus and tau k minus for all the modes. We start off with a random initialization where the motif as well as the read distributions are not well defined. Then we go on to sample and keep on sampling till the posterior probability does not increase for 5n iterations, where n is the number of sequences. Since we do not know the best value of m, we learn the models with different values of m and calculate the Bayesian information criteria or BIC score for each model. BIC depends upon k, which is the number of free parameters of the model, and the posterior score at the map estimate. K can be easily calculated from the width of the motif and the widths of the read footprints as well as the tau values. Now the best model is the one with minimum BIC score. Let us look at some of the results of applying exodiversity to different datasets. This was the result of exodiversity on Chipexo for SOX2. To get a better view of the footprints, we look at a 100 base pair region. Exodiversity divides the dataset into 12 modes of binding. The first 7 modes are variations of the SOX2 motif itself. Modes 8 to 10 are variants of OCT4 SOX2 heterodimer. These modes are also more occupied by OCT4 in its own Chipexo experiment, as shown in gray. If we look at the 5' flanking regions in the modes 1 to 7, we see that they differ slightly. Correspondingly, the positive and the negative strand read windows are also shifted by 1 or 2 positions. This indicates that these modes might be structurally different. Even though OCT4 is not binding in the modes 1 to 7, the position of the positive strand read window is similar to that in modes 8 to 10. This indicates that the exonuclease activity might be getting hindered in a similar way in both the cases. We also ran exodiversity on the OCT4 dataset. It discovers 30 modes, out of which many are variations of the OCT4 SOX2 heterodimer again. We were curious to see exodiversity finding a large number of modes in both the SOX2 and the OCT4 datasets. To ensure that this is not due to overfitting, we set up a binary classification using SOX2 and OCT4 to see how well it predicts on unseen bound data. A standard 5-fold cross-validation was set up. Exodiversity was run on the training sets of both the TFs, SOX2 and OCT4. The beauty of the model is that we can easily calculate the log likelihood scores. The held out datasets for both the TFs were then scored using the models of SOX2 and OCT4 on the log odds score. We plotted the ROC curves on the test datasets. We see that when we use both the reads and the DNA information, the AUC obtained is greater than the case when we use only the DNA information. But this is expected as the reads are indicative of the corresponding TF binding. To compare the performance of exodiversity, we use the state-of-the-art classifier GKM-SVM. It goes beyond using single PWM and uses gapped k-mers to distinguish between regulatory elements. However, the AUC obtained by GKM-SVM on the held-out datasets was still less than that obtained by exodiversity. In the precision recall curves also, we see the difference in the AUCs between the training and the test dataset is greater in case of GKM-SVM than in case of exodiversity. We ran exodiversity on the Chipexo for FOX-A1 dataset. It divided the data into 13 modes. The first 11 modes are variations of the FOX-A1 motif itself. Mode 12 is the CTCF motif. If we look at modes 1 and 2, we can see an additional C at the upstream position. Mode 2 is known to be enriched in regions bound by both FOX-A1 and FOX-A2. If we look at the positive strand reads in these two modes, they are shifted by a few positions as compared to mode 5, which is the best match to the canonical motif. Having more information content in the motifs does not always mean having more number of reads. The modes with less number of reads might be bound by another protein, and due to the competition between FOX-A1 and the other TFs, lesser instances of FOX-A1 bound regions might have been pulled down in the experiment. But we need more information to confirm this. We also ran exodiversity on the entire Chipexo dataset for CTCF. Since CTCF uses flanking motifs, we started exodiversity with an initial motif width of 40. It identifies 8 modes resembling the core CTCF motif. The positions of the positive and the negative strand read windows depend upon the flanking motifs. If we look at modes 1 and 2, we see both of them have the upstream flanking motif. However, the downstream flanking motif in mode 2 is weaker, and the negative strand read window is also shifted towards the core in mode 2. We plotted the conservation values at the modes identified by exodiversity, and we see that they correlate with the read distributions. It is reassuring to see this, as this information was not given to the model. Exodiversity was run on chip nexus for twist in Drosophila. It divides the dataset into 25 modes of binding, out of which 9 modes are variants of the twist motif itself. It also finds a core 1 promoter motif and CA repeats and GA repeats, which are characteristic of fly enhancers. We plotted the DNA shape propeller twist values at the modes identified by exodiversity, and we see variations within the twist motifs also. The modes 1, 2, 4, 6 to 8, which correspond to the CAYATGE box, have a low propeller twist signal at the center and a slightly higher signal at the upstream region, while the modes 3, 5 and 9 have a strong propeller twist signal at the center. A high propeller twist signal means more DNA rigidity. This is indicative of the differing positions of the read windows in both the strands, as we can see in modes 4 and 5. To conclude, we see that exodiversity provides a novel approach to use both the read counts and the sequence motifs to categorize and resolve the Chipexo footprints. It identifies small nucleotide level variations within the binding motifs, and they correspond to the read signals. Different modes have different characteristic features in terms of sequence conservation, DNA shape, proximity to the TSS, which we did not see here but we have discussed it in the paper. Thus, exodiversity can be applied to other types of data as well where nucleotide level information is available. Lastly, I would like to thank all the people who helped us with the datasets, the brainstorming sessions, and the discussions. I would also like to thank my funding agency and to all of you for listening. Thank you.